So, yes. Um, so today we'll be talking about the different types of extracellular matrix, or what we call as ECM. So ECM is a network, or it's a mesh of macromolecules that generally in our body in physiological condition is caused due to the different molecules or proteins or carbohydrates that are secreted by the cells, and that allows the cells to adhere to the surface and get nutrients for them. So this particular ECM, it contributes to the properties of the tissues or cells, that it is the cells like specific for the cells or tissues in which it grows. And so it depends upon which cell type, or it depends upon which part of your body, which part of the body, which part of your body the cells have been extracted from, in order to have a better idea of what ECM it would be suitable for growing those cells. Because each extracellular matrix components are different, although the basic components like the molecule, like the proteins, carbohydrates, might be similar, but there might be changes in the amino acid sequences of the proteins, or there might be changes in the carbohydrate monosaccharides, or whatever disaccharides the ECM matrix is made up of. So depending upon the requirements of the cell, depending upon where the cells are growing, the ECM can be different types. And in our in vitro conditions for cell culture, different kinds of ECM is used. One kind is naturally occurring, and the other kind is synthetically. Sorry, I have just made it opposite. The bottom one is actually natural. This is not synthetic. This is natural actually, and this is natural, and this one is synthetic. So we have different kinds of ECM that we use for coating the surface of the glass dishes, or the petri plates, or the T25 flasks, whatever we are growing our cells in. We use different kinds of ECM matrix for coating those dishes. And it depends upon what cells we are growing. So it can be, the ECM matrix can be divided into two parts. One is the synthetic ECM, and the other is the natural. So synthetic ECM consists of poly-D lysine and beta-silane. So lysine, as you know, is an amino acid. So the D isomer of lysine, when it is when many monomers of lysine, when many monomers of D lysine are conjugated together, it will form poly-D lysine, and that is synthetically made. It is not naturally occurring. Also, beta-silane, silane is made up of silica and beta, and there are different categories of how many adjacent molecules of nitrogen containing peptide sequences are attached to the silica. It depends upon that. So beta means there are three nitrogen containing peptide sequences which are added to the silica. So this is the two synthetic synthetically synthesized ECM matrices, which are used in the regular cell culture by different people for their requirements. Also, the naturally occurring ECM matrix, which many people use, are collagen, gelatin, and laminin. So all these proteins, I think some point or the other, you might have heard it in different aspects, like collagen is present in the nails, gelatin is present in like different algae. We generally use gelatin for making the the jelly kind of substances. So for that, and also laminin is present in also in nucleus for maintaining the structure of the nucleus, this laminin proteins is required. So these are naturally occurring proteins. These are present in the environment, and we don't have to chemically synthesize it. So these are naturally occurring extracellular matrices, and these are also used similar to the synthetic ECM for different parts of cell culture. So the structure of lysine, as you know, is like this is the amino acid lysine, D lysine. So I think what is the difference between D lysine and L lysine? I'll just say in brief. So the D and L part is the stereoisomers. So D and L, depending upon which direction the particular amino acid rotates the plane polarized light. So that is some concept of chemistry. So stereoisomerism depends upon, like, when we are shining a plane polarized light. Plane polarized light means light which vibrates in only one particular direction. So if the light vibrates in this particular direction, in only one direction, that is known as plane polarized light. And if this light, when it is shined on a particular amino acid sequence, an amino acid molecule, so if it rotates the light towards the right, if the light is towards the right, then it will become a D isomer, D stereoisomer. If it rotates the light towards the left, if the molecule rotates the light towards the left, then it is a L isomer. So naturally occurring amino acids are the L isomers. So D lysine is not naturally. I mean, in the bodies of humans or animals, in mammals, basically, the amino acids that are present is generally the L isomers that are naturally occurring. But in case of bacteria or the lower organisms, the D component of the amino acids are present, and that is why many times what happens is when bacteria or microbes, they attack humans. So because of their changes in their amino acid sequences on their surface, like the LPS layer, the LPS layer that is present in the gram-negative bacteria, that is made up of D isomers of amino acids. So if bacteria is attacking humans, then the immune system can recognize that is a foreign substance, because of one of the reasons is because the amino acids are in different configuration. So in bacteria, these are in D isomers generally, and in mammals or in higher organisms, the naturally occurring amino acids are L isomers. So this is the structure of D lysine, and so in D lysine we have the normal COOH and NH2CH, and the side chain is contained of another amino group which can be which can get the positive charge, which can get positive charge on it, and therefore lysine is generally a positively charged amino acid. So it's called a positively charged amino acid, and positive charge. So this is a positively charged amino acid, and lysine is so if it is positively charged, it's tend to be basic. So lysine is a basic amino acid, and it's a positively charged amino acid. So when many molecules of lysine, when many monomers of lysine join together, they form poly D lysine. Or how they form poly D lysine is they will so when two monomers are joining together, so basically if there are two monomers of lysine, so suppose I'm doing this, and then O and HOH. So it will remove water from the sequence. Suppose it will remove H2O, and it will form a peptide bond over here. So here this OH and one H from the NH2 will be removed, and here we will form a peptide bond between the two monomers of amino acid. So CONH bond that we say is the peptide bond. And as a result of which we will be getting the amino acid, the poly D lysine, which will have several sequences like this, and the NH2 plus, that is the side group. The side group NH2 plus will remain as positively charged side group. So as a result of this, the poly D lysine entirely, it will have positive charge, positive charge, positive charge everywhere. So basically, if we are coating the surface with poly D lysine, then the NH2 plus group will be protruded outside, and it will give positive charges on the outside. So any molecules which show positively charged, has positive charged positively charged side groups, they will show attraction towards negatively charged groups. So, poly D lysine, when it is coated on the surface of the substrate, so suppose this is the plate or the cell culture dish. So this is the cell culture dish, and we have polylysine coat on it. So the polylysine coat will be attracted towards the negatively charged membranes of the cells, and so then the cells will be interacting with poly D lysine through the negatively charged membrane, and as a result of which we will get the cells are adhered on the surface of the substratum. So depending upon what concentrate, because if we have too many positive charges or too little positive charges, then the cells will not have enough amount of electrostatic interaction to bind with the poly D lysine coating. So the concentration at which the cells will concentration of poly D lysine that we need to use, as a result of which the cells can adhere, will be different. So it is not just true for poly D lysine; it is true for each of the ECM that is coated. So depending upon what is the cell type, it is always not always good to say that this concentration will work. So it is different for different cell types, and depending upon the cell type that we are working on, we have to optimize at which concentration it is working, and at what how much volume do we need to actually spread on the dish, so that it is like properly spread on the dish. So those optimizations have to be done based on what cells we are working with, and how the cells are behaving. So the main principle behind poly D lysine and cells interacting or adhering is because of the electrostatic interaction. So apart from poly D lysine, another synthetic ECM that we use generally is silane. So silanes can be different types, that is EDA or DTA or PIDA. So it depends upon what is the side chain that is used. So this is the molecule which is called the silane, and depending upon what side groups we are using, they have different chemical names. So the particular DTA silane is used in case of making synthetic ECM matrix, because this shows better. This also has NH2 groups, as you can see, and so this also shows positive charges, and this is better. Like we need to have a great so we cannot. So when we are using ECM, so we cannot have very bulky side chains like this one. We cannot have that, and also we cannot have a little smaller peptides. So ECM requires that we should have a minimum length of the peptides or side chain that is attached to the main backbone, and also we should have a charged molecule, preferably, so that it can interact with the opposite charges of the cells, and it can bind to the cells, and the cells will be adhered. So then it is like we have to. So here that is why DTA silane is used for DTA silane is used as a synthetic extracellular matrix for coating the substrate, and the cells can adhere to the substrate using this. So this is the two synthetically used ECM that Sir was talking about in his lectures. So next, what he had discussed about cleaning the glass substrate properly, and what should we do before we are proceeding with coating the surface with extracellular matrix is we clean the glass substrate, or the substrate in which we want to coat with ECM. We have to clean it properly. The reason being, anything that we are getting commercially, even if they are cell culture grade, or even if they claim that it is cell culture grade, it is not totally sterile. And before plating our cells, and before performing a cell culture in those glass cover slips or glass dishes, we have to make sure that everything is clean, and there should not be any interfering substances apart from the ECM and the cells for continuing the cell culture. Now, many times what happens is, in our lab we don't coat the glass dishes, Petri dishes, 35 dishes, or those dishes, because they come as pre-coated, and they are like pre-coated and suitable for growing the cells which we grow. So, but certain cells that do not grow in the ECM that is already coated in this dishes, for those cells only we need to coat extra. Like I was discussing in the previous class, that some cells are very loosely attached to the substratum that already the company provide, like the dishes which are already coated with the substrates. So some certain cells do not attach to that substrate very tightly, so they are very loosely attached. For those cells we need to coat it, and otherwise, if the cells are growing fine in the pre-coated dishes, it is good to go, and it is fine that we don't need any extra coating. But if we do need an extra coating, then we have to make sure that whatever substrate we want to coat the ECM matrix with has to be totally clean and free of oil, free of other residual dust particles. So it is very common that there is a lot of dust particles present in the cover slips or in the Petri dishes, which is not visible to the eyes, but it is present. So we have to be very careful in removing that. So first and foremost, we have to clean it with soap water. Soap water is just normal soap water. You can use Vim bar, you can use also the normal Dettol, but also another thing that I is keeping it overnight, because soap also has soap has a lot of salts. So we don't want salts to interfere with the ECM. So just one wash with soap, and then properly rinsing it with proper water, not the tap water, but the milieu water or the distilled water, which is clean of all the other salts or elements. So we have to, when we are washing especially the cell culture equipments, we have to be very careful to not use water from the tap, because tap water also has a lot of contaminating microbes, and apart from that, we also have a lot of salts. So we don't want any salt interference. So we have to first, when we are cleaning it with soap water, and then we have to rinse it properly with normal water. After rinsing it with normal water, before coating, we need to use concentrated nitric acid for etching the surface. So what do we mean by etching the surfaces? So generally the glass, the first layer of glass is it's not very like uniform. So etching the surface will make it little more rough, and if we are making it more rough, then what happens is, when we are coating the ECM, it will be properly so if it is too smooth, then the ECM will slide away. But if it is little rough, if the surface is little rough and has little grooves in it, then the ECM can be when we are pouring. So ECM is not a solid material, it's like liquid only when we are pouring it. So it will be properly spread in the surface and can be properly coated. So we need to make, so there should be little bit friction so that it is spread properly. Otherwise it is not going to be uniformly spread. So we want to etch the surface, and so we use certain corrosive agents, and generally people use concentrated nitric acid for etching the surface or making it rough by removing thin layers of surface material. So the initial the surface material, like thin layers of surface material, is removed or it is corroded using this concentrated nitric acid. And then after etching the surface or making it rough, then we wash it again thoroughly with water to remove the residual acid. So after every step, it is very important and crucial that we have to wash it with water, because any of these things, the soap water or the acid, or after that we have to use ethanol also, so any of these things are not very growth friendly for the cells, because these are very toxic. And these are very toxic, and this leads to, I mean, if it is there in the cover slip or if it is not properly washed off, then the cells will die. So every time when we are washing, we have to be very careful that there should not be any residual soap water, there should not be any residual acid or any residual alcohol in the dishes that we are going to grow the cells in. So yes, so after washing with soap water and then etching the surface with nitric acid, we have to wash it with again water to thoroughly have to wash it so that we remove all the residual acid and the soap water and whatever. And then we have to pour 100% alcohol. So why do we pour 100% alcohol is to remove contamination basically. So 100% alcohol, it's a disinfectant, and during cell culture alcohol has to be your best friend, because it is very essential that whenever we are performing cell culture, we need to properly wipe our hands with alcohol, wipe the desk with alcohol, the hood with alcohol, everywhere, wherever you put yourself, there has to be alcohol. So if you are going to grow the cells in the cover slips, so cover slips, these are very thin cover slips, so they need to be handled with the help of forceps. So we have to take the cover slips and then we have to put it in say a small dish, and put alcohol on the dish and put all the cover slips in the dish and let it be for some time. Like it should be totally immersed in alcohol, so that whatever microbes the cover slips will have, or whatever microbes the dish will have, it will be removed. So then after properly soaking it with ethanol, then we have to burn the cover slips. Now we don't need to put water again. Once we have put ethanol, is it can be evaporated. So ethanol evaporates. So before it gets evaporated, we have to burn the cover slips in the flame, so that again the residual microbes are removed and also the ethanol is totally evaporated. So just we don't need to place it on top of the flame, because if you are placing it on top of the flame, there is chances that it will break. So it has to be little far from the flame, and we can see that when we are burning that ethanol, when we are burning the cover slips or the dish, the ethanol will start evaporating because of the heat itself. So just from far we need to place the cover slips or the dish in front of the flame, and the residual ethanol will be removed. So once the residual ethanol is removed, then the cover slips can be can be plated in sterile cell culture dishes, and the rest of the cover slips that so basically while performing this, whole cover slip bunch should be going through the same procedure of cleaning with soap water and then etching the surface and then putting alcohol on this, and just we can put it in alcohol and parafilm it and then keep it for the next set of experiments. And only take out the cover slips in the hood itself. Once it is in 100% ethanol, we have to place it in the hood, in sterile cell culture hood, and place it under paraffin wrapping, and only take out when we require for the next set. We should not keep it open, and we should not keep it outside the hood. to reduce contamination issues. So yes, so the cover slip that we require for experiment, then after burning it in flame, we can plate it in cell culture dishes. So in the cell culture dishes, we can plate our cover slips or slides or whatever we want to do. So this step is similar for cover slips, glass slides, or whatever dishes we require for coating the ECM on. So once we have done this, we can put it in sterile cell culture dishes, and then we can proceed with the coating with suitable ECM. So all these ECM metrics that is talked about, the poly D lysine or the beta-silane or collagen or gelatin, whatever. So we get it in bottles, and those bottles can be stored in, depending upon its storage, it's -20 or room temperature or whatever the storage is. And we have to dilute it in concentrations depending upon what solvent it gets dissolved in. So if it gets dissolved in some water aqueous solvent, then we have to use water. If it is dissolved in some ether or some polar solvent, then we have to, depending upon what is the requirement, it is always written on the bottle that if it is not written on the bottle, we have to go to the website from which the chemical was brought, and then we have to see in what solvent it gets dissolved, and then we have to dissolve it in autoclaved molecule system, like autoclaved molecule water or ether. So all those things have to be autoclaved before dissolving it, because even the ECM is going to go in the cell culture use. So whatever we are using for cell culture needs to be properly sterilized before using it in cell culture. So yes, once we are done with all the cleaning process and all the etching, cleaning, and everything, and once we have plated it on the dish, then what I generally do is I put it in UV for 20 to 30 minutes. All the cover slips which have been now cleaned, and we are putting it in UV for 20 to 30 minutes, and then I will come and then I will seed the cells. So UV also will show surface sterilization and will remove any residual microbes which are present. So once this is done, then we coat it. Now we have to check which ECM is suitable for growth. Like whatever ECM, generally very commonly used ECM is poly D lysine. And depending upon other cells requirement, other ECM could also be used. But poly D lysine is a very commonly used ECM, and many people use it. So we have to check which concentration of ECM is suitable for cell growth, because not all concentration will promote cell growth. If the concentration is very high, it might restrict the growth of the cells, and cells might be stagnant and it will not grow. Also, if it is lower in concentration, the cells will not get enough ECM matrix to adhere on, and it will be floating or rolling on the surface. So that also we don't want. So we need to be very we have to optimize the concentration in which concentration of ECM the cells are growing. Once the concentration is standardized, so we can use different concentrations of ECM, so 1 mg per ml, 2 mg, 3 mg, and then see in what concentration it is perfectly working. Once that is standardized, then we have to see that whether the substrate that has been coated shows physical, what is the what are the different nature of the surface which is coated with ECM. So depending upon how much the surface is rigid or how much the surface is rough, we can detect it using AFM. So AFM is known as atomic force microscopy. So atomic force microscopy helps in detecting very minute changes in the surface of the on the surface, like on the surface of anything. So basically the physical surface analysis, or how the surface is, basically if it is rough, if it is very much rough, or if it is smooth surface, or if it is kind of very peaky, like kind of like very pokey surface. So we have to make sure that the surface is not very rough or very smooth. So it has to be somewhere in the middle. So that we can confirm using AFM. So AFM is atomic force microscopy. And in atomic force microscopy, the principle is we have a small cantilever. So this is called a cantilever, and this is attached to one rod-like substance. So what happens is light shines on the top of this on the top of the cantilever, and it can reflect the light based on how much it is dipping inside or outside. So basically, if the surface is like this, then your cantilever is going to it can detect the surface by this region, and in the needle region of the cantilever can detect how much the surface is deep or how much the surface is elevated. So if it is deep, then the cantilever will go down, and so as the cantilever goes down, then the light that is shined on the cantilever will be reflected in a different angle. Once it is going up like this, so it will be coming up like this, and then the light will be shining in different angle. So we can so on the movement of the cantilever on the surface of the dish or the cover slip, we can get a pattern like how the surface is. So we can see whether the surface is smooth or rough, or what is the degree of smoothness or what is the degree of roughness using this technique. So this is a very sophisticated technique, and atomic force microscopy is very much it is very much affected with sound noises or even the vibration. So generally this kind of microscopes are placed in the ground floor or even better underground, because the building vibration that we have, if the building is very tall, so the building vibration can also affect the movement of cantilever. So this kind of microscopes have to be placed in a very secure room, preferably in the ground floor or underground floor, so that the building vibration is not affecting it, because it is very sensitive. It is measuring the surface dynamics, and it can even detect DNA you can see DNA using this technique. So it is very sensitive, and it is very much prone to getting affected by the vibration outside. So so physical surface analysis is performed by this kind of microscope. And then next we perform contact angle measurements. So what do we mean by contact angle measurements? So whenever we have a surface which is coated with any kind of ECM, so if the surface is very smoothly coated, then if we are putting our media, or if we are putting buffer on it, the media or buffer will be spread, and it will not be in 90-degree angle with the substrate. So basically, if it is in 90-degree angle with the substrate, so if the angle is 90-degree, then it is not properly, the ECM matrix is not properly spread. If the matrix is properly spread, then the contact angle here means the angle with the media or with the buffer, whatever we want to put on top of the ECM, media or buffer containing the cells. So if our media or if our buffer containing the cells is properly spread around, then the contact angle will decrease, and will decrease from 90-degree to less than 90-degree. So this will become less than 90-degree. Rather, if the so this angle is less than 90-degree, but if it is not properly spread, then the angle will be around 90-degree. So better, so if it is properly spread, it will be less than 90-degree. So better cell adhesion will be achieved when the ECM is properly spread. So this is better way of if your contact angle is less than 90-degree, that means it is good to go. I mean, it is lesser contact angle means good spreading of ECM, and that is good for the cells to be adhered and to grow on it. Also, it can different. So this differentiates between the hydrophilic and hydrophobic nature of the surface. So basically, if it is more hydrophilic, so it will attract water or media. So generally, the cells are grown in media or buffer, which is water based or which is aqueous phase. So we need to have an hydrophilic nature of the surface. If it is more hydrophobic, that means the ECM is not a proper choice, and we need to change the ECM. So more hydrophilic nature of the surface will promote the cell adhesion. Also, we need to do surface chemical analysis. So yeah, so these are the three different things that we need to perform before doing cell culture. But this is not, this is not very much practical, in the sense that not every institute will have such sophisticated techniques like AFM, or measuring contact angle, or doing the surface chemical analysis. These are generally done once, or maybe these have been done like initially while standardizing, but after that, later on, people have been using the same thing again and again, and same concentration. So different labs will have different protocols for using an ECM matrix, and different cells. So depending upon the cell that the lab works on, so these are more or less standardized. But the initial method for how we need to actually standardize the concentration, how we need to actually see whether the surface properties have been changed a lot or not, these are the techniques. So after measuring the physical, after seeing the physical surface, after doing the physical surface analysis, and after doing the contact angle measurements, this is done using goniometer. And after that, we need to do surface chemical analysis. So basically, surface chemical analysis will tell us whether the surface has a certain kind of atom or not. So depending upon whether you're using carbon-based ECM, whether you're using silica-based ECM, whether you're using nitrogen-based ECM, the XPS means X-ray photoelectron spectroscopy. So this is again a technique which can, detect the surface atoms and how much quantity they are present in, and they can be we can be sure that okay, this our ECM contains silica more, or our ECM has more carbon in it. So depending upon that, the surface chemical analysis can be performed, and whatever cells we are growing on, and we can optimize the ECM concentration or the ECM composition accordingly. So here it is, so X-ray photoelectron spectroscopy. So it's a surface sensitive, not bulk. Surface sensitive means, suppose your entire dish, suppose this is the dish, and this surface has different kinds of atoms on it. Also, it will be present in the bottom, but we are not interested in the bottom phenomena. We are not interested in the bulk, but we are interested in what happens in the surface. So X-ray photoelectron spectroscopy is a surface sensitive technique, and it can detect both qualitative as well as quantitative measure of how much and what kind of atoms are present. So suppose there is different kinds like carbon or nitrogen or silica or chlorine or whatever. So whatever we have, depending upon whatever surface, whatever atoms we have on the surface, so depending upon that, there is it basically works on the principle that the kinetic energy of electrons after getting excited from different atoms will be different for different atoms. Basically, suppose there are different number of electrons present on the outer shell in different atoms. So depending upon the number of electrons, depending upon the size of the atom, the kinetic energy at which the electrons are excited, it will be different. And we can measure the speed at which the electrons are coming out. So this was later on described as the photoelectric effect, or the photoelectric effect of the electrons have been measured using this technique. So the photoelectric effect will be different for different kinds of atom. So whenever we are shining a X-ray beam, so this X-ray beam, on this, suppose this is the sample and has different categories of atom. So this whenever it shines, the electrons will be excited out with different, ejected out with different speeds. So this speed of the electron energy, kinetic energy of the electrons, of the photo emitted electrons, only from the top surface, not from the entire bulk surface, just from the top surface. So only from this surface the electrons energy is being measured, and this measures the kinetic energy of the electrons, and also electron detector will count the electrons, and it will show, so this is the detector, and it will show signals as peak. So it will show this kind of peaks, and these peaks are kind of standard peaks for certain atoms. So for example, oxygen will have a certain kind of peak, nitrogen will have a certain peak, carbon will have a certain peak. So depending upon the peak nature, and depending upon depending upon the peak nature, we can detect that what our surface mainly consists of, and whether the surface has the desired atom that we want to or not. So depending upon that, so this kind of technique is used to detect the atoms present on the surface and to what extent they are present. Yes. So next, once we are done with the ECM, like once we are checking the concentration of ECM and then checking the surface properties, the physical surface analysis, the contact angle measurements, and the surface chemical analysis, then finally we have to coat the surface with ECM. And once the desired ECM is coated on the surface and it's checked using the XPS and contact angle measurement, so first set of interactions between the ECM and the cell is electrostatic interaction. So this is the substrate. It has our ECM in it, and this is our cells. So these cells will interact with the surface using certain electrostatic interaction. So this is the first set of interaction that is established between the ECM and the cells. And this is mainly because the ECM consists of some charged particles which can interact with the opposite charge of the cells. So this first set of interaction between ECM and the cells will allow the initial adhering of the cells with the ECM. So that will be the initial binding force that will cause the cells to get attached to the substrate. Once the cell is attached to the substrate, the cell itself will start secreting ECM of its own, and this ECM can now that the cell secretes ECM of its own, more and more cells which were not initially bound to, they will also start getting attached to the substrate. And they will also get attached to the substrate and they will get adhered. So now, depending upon our requirements, now whether we want to culture the cells in 2-dimensional or whether we want to culture the cells on 3-dimensional, we can put a thin or thick layer of ECM. So generally for 2D culture, so mainly people do 2D cultures for microscopic experiments or immunohistochemistry or any kind of other molecular biology experiments like PCR, Western blotting. So cells for those experiments can be coated in 2D, and we work with 2D cell cultures. But if we want to see characteristics like cell migration, or if we want to see characteristics like what kind of cells, like how the cells are in physiological condition and how the drugs are affecting the cells, then cells can be grown in a 3D environment. And in 3D environment, the ECM that needs to be spread on the surface of the substrate needs to be thicker, and that thickness is not common, or also is depends on what cell type we are working on, and it should be properly analyzed, like how much the ECM should be there so that it is porous enough for the cells. So keeping the porosity in mind. So how much porous the cells, the ECM needs to be so that the cells can be accommodated. So basically, if the ECM is thick, so if the ECM is thinner, there is no such problem, because the cells will only interact with this region of only the lower portion of the cell's entire structure. They will only interact with the lower portion. But if it is in a 3D culture, so if the cells in a 3D culture, that means the matrix that is present has to be interacted with all the three-dimensional regions of the cell. So the cell here is in the bottom, so in the middle, and it can interact with the ECM with all different direction. And so we have to design the ECM in such a manner that it is not too congested, and it does not have space for the cells to be accommodate inside it. So we should have so that is what is known as porosity. Porosity means we should basically when we are making any gel, how much tightly the molecules are packed inside the gel, that will determine the porosity. So we need to have an optimized porosity level so that the cells that we want to grow gets accommodated in the system. So once we are done, so the porosity of the system can be measured by the instrument porosimeter, and we can the porosity of the system also needs to be maintained so that there can be proper air diffusion and air exit, because the cells needs to grow as well. So the ECM matrix should not be very opaque, or should be breathable, so that it can allow the cells to grow in it, and also it should be able to accommodate the cells. So depending upon all these requirements, whether it is how much porous it is, how much surface rigidity is there, roughness is there, and what chemicals are used, and this and that, finally we coat with the different kinds of ECM, and the natural, so Poly-D Lysine and deposition. So Poly-D Lysine and deposition. So these are the synthetic ECM matrix, whereas the natural matrix consists of different laminin, gelatin, and concanavalin A. So this is these materials are naturally occurring, and it is present in different cell organelles or different animals as well. So we can isolate it from there, and we don't need to chemically synthesize it. Now, whatever cells we want to grow, depending upon the requirement, this kind of ECM is required. So laminin, it is a naturally occurring ECM, and but it is very difficult to coat on the surface of glass. So laminin cannot be directly coated on the surface of the glass. So this is the glass, suppose, and laminin cannot be directly coated because it does not interact with glass much. So this laminin, in order to properly coat it, we have to coat the glass first with ornithine. So this has to be coated with ornithine first, and once ornithine binds to the glass, then so this is the glass, and here ornithine will bind, and once the ornithine is coated, we can coat the laminin. So ornithine helps coating the laminin on the surface. Now, ornithine is not a naturally occurring ECM; it's a synthetic ECM, but it allows the laminin to be coated. Once the laminin is coated, then it will interact with the integrin receptors of the cells. So cells have different proteins that helps in cell adhesion. Okay, there is adhesin, there is integrin. So integrin is another protein that helps in cell adhesion, and with the integrin receptors, laminin will interact. So suppose this is the cell, and this cell has the integrin receptors, and these integrin receptors will interact with the laminin, and so the cells will get attached to the substratum. So this is the way how a laminin helps in interacting with the cells. Now another kind of naturally occurring ECM is gelatin, which is a mixture of laminin and poly D lysine. Now poly D lysine is we have to synthetically make it, but the mixture of laminin and poly D lysine is gelatin, and that we get it naturally. And another molecule is concanavalin A. So concanavalin A, it belongs to the family of lectins. So lectins are carbohydrate binding proteins. So these are also present on the surface of so this concanavalin A, when it is coated on the ECM, like when the ECM considers concanavalin A, it can bind to mainly rods and cones. So these are rods and cones present in optic sensation. So this mainly binds to the cells of this. So depending upon the cell type, concanavalin A, which is a carbohydrate binding protein, and it is present on this ECM, so it will bind to this kind of cells, yes. So this allows the attachment of rods and cones. This is concanavalin A, and this binds to rods and cones. So yes, so this is all about natural ECM. Now maybe we can discuss some questions, or maybe we can go into some doubts, and then we'll go to questions. So anybody has any questions so far?
Ma'am, is ECM present only in animal tissues or both in plant and animal tissues?
Can you speak little loudly?
My question is that is ECM present only in animal tissues or is it present even in plant tissues also?
It is present in both plants and animals, both.
But when I did plant tissue culture, but we don't use any ECM over there.
Yes, because generally we also don't, even in animal cell culture, also in our lab also, we are not using, we are not coating it externally. Whatever is pre-coated, maybe the dishes or maybe the vessel where you are culturing it, it might be pre-coated. So we don't need to extra coat it. Only this is required for certain kinds of cells which are very fragile. That is what I have realized. But in the body, like inside the physiological condition, ECM needs to be present. So Trishla is asking, how does the higher concentration of poly D lysine affect cell growth? So poly D lysine is not a naturally occurring substance, so it's a synthetic molecule. So I think if there is more amount of poly D lysine, that will require more, that will have more amount of positive charges, and that will maybe it will not show like it will maybe interact with the cell so much that the cell cycle is arrested in some way. I don't know about the mechanism behind it, but yeah, maybe it will become toxic to the cells and cells will die.
Yeah. I have one question. For example, I have a flask which was taken from the external vendor, and so ECM is available on it, and I need to add a culture on it. So, for example, if I want to add a 10 ml of culture, instead of that, I added a 20 ml. So, what will the effect on cell growth? So, how it will be affect the this proliferation?
So if the culture vessel, so it will be having certain amount. So suppose in case of certain culture vessel, we can not like exceed the amount of cells that we want to seed. Only what will happen is your cells will grow very fast. I mean, suppose you're adding 10 ml instead of 5 ml, it's not going to affect the growth as such. I don't think so. But if you're like, because already you have a lot of cells there, so your cells will become confluent more quickly as compared to what would have had what would be in case of 5 ml culture. And also, if that happens, so overconfluency causes stress to the cells, and stress to the cells in the sense that if the cells are too much and the space is very less, then the cells will not have enough space to grow, and so it will start. I mean, the cells will start dying. So the stress will cause cell death. I don't think so. ECM will have any effect there, because already if you are buying already if the ECM is suitable for the cells that you are growing, so it's not like the ratio of ECM is to your cells are going to be affected, just that less number of cells will get adhered. But already if you're adding more number of cells, then we have we don't need to worry about whether cells are getting adhered or not. But yes, cells will get confluent more fast, because more amount of cells are present as compared to your substrate. So it will get overconfident and it will be stressed. So it is not so we have to be very careful with how much amount of cells we have to seed, or how much, because we cannot seed less amount of cell also, or we cannot seed more amount of cell. If we are seeding less amount of cell, then what will happen is the cells are so less that the cell to cell contact is also not being able to maintain, as a result of which also cell will get stressed. And also if the cells are too much overconfident, then also the cells will not have enough space to grow, and then also the cell will get stressed. So it is very important that we have to maintain that ideal concentration, or the ideal volume in which we want to seed the cells.
Perfect.
Okay, so Bharat is asking, explain about synthetic ECM. So synthetic ECM is anything with so as I discussed, like poly D lysine or this D-Lysine. So these are not naturally occurring proteins, or these are not naturally occurring peptides. So these have to be synthesized externally in the laboratories or by companies. So those are synthetic ECM, which is which are synthesized and not naturally occurring. Naturally occurring ECM is which is present in the cells or in the animals. We have to just isolate it. We have to purify it. We don't have to chemically synthesize it. I actually had a doubt. So Gunjan is asking, week 3. I just wanted to know if it's the right time. Week 3 assignment. So you have doubt regarding question, or did you get the answer? Because I don't know whether I'm allowed to discuss the answers if it is not already or did you get the answer for the week 3 assignment? Like, did you already get the marks for it? Something? Revant, okay, so I will actually, I had talked about the potency of the cells, yes. So I will upload those videos and this thing also, my PPT also. So when do you have the exam? Okay, on October 28th. Yeah, yeah. So I will upload it. I will upload it this week only for sure, and then you can go through it again because I'm very sorry. I don't know why I was not getting any time. So I will upload it again. So you will be able to see all the slides and all the these things recorded version again. So this week only, I'm for sure I will upload it so you can see it again. So I got the answers, but I think there is a misunderstanding regarding one question. So, okay, what is the question? Autoclave tape, their sterility. This is autoclave. Okay, this is the fill in the blanks. So it assures sterility and indicates that proper dash was reached during the process. Proper. Mm, is it autoclave tape or is it just autoclave? It will be autoclave pressure, no? It has to be. Okay. So yeah, it has to be, I think, fast only. Assure sterility indicates proper pressure was reached during the process. It should have been inverted upright. Uh, which is inverted upright? I mean, which? Can you tell me the question, Gunjan? Let me see. Okay. So, question is source of light and condenser for dash is placed below the stage and is placed above the stage. The source of light and condenser for uh, inverted. No, it is. So yeah, it is. So the source of light and condenser. The inverted so for upright microscope we have so upright microscope is from the inverted microscope from top. So from top the source and for inverted microscope, for upright microscope has to be placed below, and for inverted above the stage. So upright and inverted, I think it is. What is the answer? So yeah, the source of light and condenser. So wait, I will open my previous. So for the upright microscope has to be placed below. I mean, wait. Let me share the screen. So here, this is the upright microscope. Okay, so for upright microscope, it will be below the light, and for so this is inverted microscope. So for inverted microscope, it will be above. The light is from here. So the answer should be. So for upright microscope is below the stage, and inverted microscope is placed above the stage. Yeah, ma'am, but according to what sir explained in his video lecture, the answer would be third option, which has been given incorrect. So third option is actually incorrect only. I don't know what sir has said, but upright microscope only it is the light source is below the stage, and inverted microscope it is above the stage. So yeah. So there is one more question that he was asking, Revant was asking. So autoclave tape assures sterility and indicates that proper. Okay, so autoclave tape and autoclave is, I think, two different things. So autoclave tape is just to put the all your vessels inside that packet should be properly secured. I think it is just for that, not for like whether you put autoclave tape or whether you put rubber band. It is the same thing. It is not going to affect the sterility. So it will not assure the sterility. So I was confused between autoclave tape or whether it is autoclave. So if it is autoclave tape, then it is not associated with sterility, because autoclave tape is just the sole purpose of autoclave tape is to put the autoclave bag which contains your things, proper things in, like it should not be open, that's it. And also proper temperature was reached, because like just autoclave like temperature will anyway reach in, like when if we are closing the autoclave properly, if we are properly closing the autoclave, so does not assure sterility and indicates that proper temperature was reached during the process. So if the autoclave is properly closed, so irrespective of whether you're using autoclave tape or whether you're using rubber band, temperature will be reached. I think, I don't know, what is the correct answer for this? So it does not assure sterility, but it indicates that proper pressure was reached during the process. I think it will be proper pressure. I don't know. Maybe it is option number 2.
No, the which you are saying is correct. So autoclave doesn't give assurance that all the materials are autoclave. It's just an indication or to represent ourselves like okay, the temperature has reached. But we have to means make sure that by seeing the reports or by seeing the temperature sensor and everything, we can get to know that the autoclaves are perfectly done. But these give only the assurance, not in doesn't give a exactly 100% assurance. This is our personal indication. That's it. The answer will be that it doesn't give the assurance, but um, uh, for indication. And actually, the question is
So
like,
doesn't assure, but that is there. So it does not
yeah,
assure sterility. But then
yes,
what will be the like the next? But it indicates that proper what is reached, pressure or temperature, because
temperature change.
temperature, if we are closing the autoclave, so if we are closing the autoclave and if we are switching it on, so if the pressure is reaching, then like suppose the pressure has reached and temperature is so the if we can get the indication that the pressure and temperature is reached in the outside only. So how we will be like, so whatever is inside the autoclave should reach the temperature.
No, see, the tape having a certain chemical, so it can, for example, before autoclave, the color of tape will be whitish color. So once the temperatures reach the 121 degrees centigrade, so what will happen? It will change the color to the brown. So the changing of
Okay.
color can be done at that temperature itself. It's not on the pressure.
Okay, okay, okay. That I'm not aware of because we use rubber bands only for
Yeah, yeah.
autoclave. So yes, okay. So then that you have got the answer, Devant. That is, it will not assure sterility, but it will assure the temperature is reached. Okay, thank you, Subhash. Um, okay. So any more questions? Okay. So now let us go into the questions. Okay. Okay, so only two of you have answered. So no, the correct answer is not 1. It has two nitrogen, because we have already 1 nitrogen in the backbone, like NH2, and also the side chain also has another nitrogen, like we had already shown. So basically we have NH2 here and also NH2 here, so 2 nitrogen in 1 molecule of lysine. So 1 monomer of polylysine has option 2 is correct. So yeah. Next question. Yes, that's correct. So option number B, anionic, because polydilysine, it gives positive charge, and so it will attract the negative charges. So it shows anionic interactions with the cells. Now the next question. So yeah, so most of you have given the correct answer. The correct answer is option A, because all the L isomers, as I was talking, mostly the L isomers of the amino acids are naturally occurring. So poly L lysine is also naturally occurring, and it gets degraded by the cell proteases. So it's not a good option for an extracellular matrix. So yes, not option A, because it's not synthetic. So it's naturally occurring. So option B is correct. So now next question. So I just showed you the structure of detasylene. So how many nitrogen is present in detasylene? Yes, so it has 3 nitrogen. So like we saw it in the structure, detasylene has 3 nitrogen, and so yeah, so the correct option is option number C. So now next question. No, C is not the correct answer. So glass cover slips, if we are washing with acid, it causes etching, and that results in like better spreading of the, the ECM. So what will happen to the contact angle then? So what is contact angle? Again, just I will tell. So if your material is not spreading and if it is just present on just one dot on the surface, then it is having a contact angle of 90°, which is not good. But if it is spreading properly, like if you are putting the like if you're putting the ECM and if it is spreading properly, then the contact angle will decrease. I mean, it is not 90°, but it is spread now, so it will be less than 90°. So that is good. So if we are washing with acid, then this causes etching on the surface, and so it will allow better spreading of the ECM. So the contact angle what should the contact angle be? So option B is correct here. So it will decrease the contact angle because it will allow more ECM to be spread on the surface. So the angle will be decreased from 90° to less than 90°. So the correct option is not A or D. So it's B actually. So yeah. Now next question. Yes, so the correct option is option number A. So integrine, it interacts with laminin, and so on the cell surface it will interact with integrine, but on the glass surface laminin interacts with ornithine to properly get attached to the glass surface, but on the cell surface it interacts with integrine. The correct option is option number A. Now let's go to next question. Okay, so the correct option for this is option number B, hydrofluoric acid, because hydrofluoric acid it replaces, so H glass means glass has silica, and this silica's oxygen, no, this oxygen is replaced with fluorine. So because it replaces oxygen with fluorine, so uh, it will corrode the surface of glass more as compared to the other acids. So hydrogen peroxide is not going to affect because it's not an acid, but hydrochloric acid and sulfuric acid, they do not interact with glass. They don't react with glass because yeah, they don't react with glass. But hydrofluoric acid, the fluorine, it reacts with oxygen and it removes oxygen, replaces this oxygen. So it will etch the glass at the highest rate. So option number B is correct. Okay, not D. Sulfur, so it has something to do with the electron, like the and the outermost shell, and then we like in chemistry classes in long back in class 12 we used to study that which element is which element can replace which element, like those things. So here fluorine is more electronegative as compared to oxygen, so it is going to replace it. So now next question. Yes, so the correct option is option number C. So we should coat it in a BSL wood under sterile conditions, because anything used for cell culture has to be very sterile. So option C is correct. Now let's go to the last question. Yes, so the correct option is option number C. Laminin, collagen, and uh, concanavalin. These are all naturally occurring ECM. Only ornithine is the odd one out, so ornithine is the correct option. So yes, that is all for today. Let us discuss any questions. Yes, so this week I will upload all the videos and I will upload all the notes. So notes in the sense of PPT that I made. So I will upload all this. You can prepare for your exam. You'll have more than 1 month. This is sufficient. So are all ECM proteins? Yes, mostly these are all ECM proteins only, because your nails and these are also kind of mostly ECM proteins only. So yeah, collagen, gelatin, these are ECM proteins. So, oh, sorry, are all ECMs protein? No. So, no, all ECMs are not just proteins. So, it's a mixture of carbohydrates, proteins, glycoproteins. So it's a mesh. It is a network of proteins and carbohydrates. It's just not proteins. Okay. Any more questions? Otherwise, we'll meet again next week with again new live session.